Our next speaker will be Anis Rahman, and he's going to speak about Cameraless Terahertz Nanometrology, an essential tool for early stage detection of skin cancer. And he's from Applied Research in Photonics, Inc., USA. Thank you very much.
Good afternoon, everybody. Can you hear me?
Yes, we can. Great.
I got to turn this thing off. Echo. So, okay. Before I start the talk, may I propose a moment of silence for all the people worldwide who died out of this COVID-19. Thank you. So as Dr. Heffron mentioned, that I'm going to be speaking about Cameraless Terahertz Nanometrology, which is an essential tool for early stage detection of skin cancer. So here is a brief plan of my talk, which is intensity-based reconstructive imaging, which illuminates the camera. And then we'll go through some of the cancer, skin cancer particularly, imaging and other way of diagnosing. And also the other part, or imaging of Stratum Corneum to quantify the interaction of protein. So this is a one slide about our company. We have been doing this terahertz technology since company was founded in 2003. And we went through these small steps over the many years. And eventually we were able to achieve lattice resolution imaging with bigger wavelength, which is terahertz wavelength. Can you see the whole screen, or is some of the thing cutting? Is this okay? Good. Okay, so I'm gonna be zooming through some of the slides, but if you have any question, feel free to ask. The imaging is a modality that's widely used in just about in any part of the science and technology or even daily life these days. However, as far as scientific imaging is concerned, there is a thing called Abbe diffraction limit, which in 1873, Ernest Abbe established that the smallest object you can image is half the wavelength of the light that you are using to image that object. So because of that, if we have to see small objects such as human cell or material lattice, crystalline lattice, or arrangement of atoms in any material, then only option we are left with is electron microscopes. So ever since 1873, it has been a big challenge that why can we not image a smaller object with bigger wavelength? So it is a fundamental limitation of the imaging technique. But yeah, as you know, people always want to overcome what is the challenge. So many efforts have been out there. We happen to be a small entity here in the USA to be able to overcome that. And I will say how. So it requires overcoming the Abbe diffraction limit, meaning that you should not be limited by the diffraction related distortion when imaging small things, such as atomic resolution. So the other thing of imaging is that all the image modalities that we have, we can see only one surface at a time. So if we want to see deeper, then there is ultrasonogram and there is X-ray. But the ultrasonogram has a resolution limit, so one can never see atomic resolution image. And the X-ray is, of course, ionizing radiation, therefore it entails radiation dose. So, and also the electron microscopes, even if you can see smaller object like atomic lattice, the high energy electron beam perturbs and distorts the actual object. So challenge remains that use bigger wavelength, see undisturbed image, see deeper without breaking it. And that's where the terahertz comes into play, that terahertz can penetrate a lot of things. Is in the middle of, as you can see here on this slide. So terahertz is in this area. Oops, let me go back there. Pen. So here's the terahertz range, which is between the infrared and microwave. A few years ago, people used to call it terahertz gap because there is no man-made source in that range. Advantage of terahertz is that it's non-ionizing and yet it can penetrate almost all material. Therefore, you can see underneath in non-destructive way. So the way we generate terahertz is simple, as this cartoon shows here. We have a nanomaterial, what we call is a dendrimer, which is a polymeric nanomaterial. Once we dope that dendrimer with a chromophore, then we can excite the dipoles with another wavelength, and that gives us the terahertz. So there are other methods of terahertz generation. Legacy technique is photoconductor or photo switch, which is exclusively being used everywhere today. But it has challenges. Biggest one is the power. Cannot be, still in the range of microwatts of power, and the terahertz bandwidth is only a few terahertz. So as I mentioned, we use this dendrimer dipole excitation technique, and I will say a few words about that. It has advantages that it does not require a femtosecond pulse laser. It does not require high voltage like the photo switch. It is tunable both in power and the bandwidth. But challenge is that it's a new technology, so most people don't know about it yet. acceptability is an issue. And I mentioned we have been doing this since 2003, so for a few years we have been around. At one point, had the opportunity to interact with Dr. Robert Curl, which is a Nobel laureate for nanotechnology or buckyballs, as well as Harry Croteau, as you can see in this picture. And in a personal note, Bob Curl mentioned that everyone knows a beta terahertz source important for scientific progress. In fact, Bob Curl's group was one of the very first group to generate terahertz radiation from technique called difference frequency from two different lasers. They could generate terahertz, but the difficulty was this one is nanowatts of power, so no practical application was possible. So what we do here is this, as I said, this dendrimer molecule, which is a polymer, but unlike a regular polymer, this is a spherical shaped polymer, like a soccer ball. But also, it can be smaller and bigger. So you can have a smaller soccer ball or a bigger soccer ball, etc. Several generations. The most attractive thing about terahertz generation is that you can attach charge centers such as chromophore molecules to this dendrimer molecule. And then that creates a dipole population. In inorganic, the dipole moment is fixed by its lattice. So not much one can do, but as you can see in this equation on the top left, terahertz power depend on two factors. One is the second order susceptibility or Chi2 of a material. And the other one is the engineering parameter, as we call it, the pump power. So one can play with pump power only so much, because unless the material has the ability to convert the wavelengths from one into another, very high power is not possible. So when we dope the dendrimer with a suitable chromophore, we can control the dipole density. We can increase it significantly. Therefore, we can get higher conversion rate as well as high power terahertz output. The other thing is that we get a broad band, meaning that not a single wavelength, but a collection of wavelength of terahertz, simply because of the fact that the dipole distance, as you know, the dipole moment is given by the charge time the separation between a positive and a negative charge. And because of the structure of the dendrimer molecule, the separation is also a function of the position, meaning that it's not like in organic lattice, they're the same distance everywhere. So because of this length, separation length between two charges, which becomes a function of the position, then the dipole moment is also a function of x. What it means that when you excite the dipole, because of the dipole population has various dipole moments, so you get a distribution of wavelengths in the terahertz range. So, and we use this terahertz source to make our instruments, such as a spectrometer and cameraless imaging system. So that is the basic technology. We have been working on it for a while. So how does one generate image without a camera? If you consider this diagram, what happens in a camera is that we have a focusing lens which focuses an object onto a focal plane array such as a CCD. And then the CCD will give out signal. That signal is treated by an onboard computer or ASIC, and that generates the image. Now, in case, I mean, in a sense, this is also a reconstructive image other than the photographic film, because you're getting a signal which is manipulated into an image. Now, the difficulty with CCD-based imaging is that your image resolution is fixed by the CCD. If CCD is a, let's say 30 megapixel CCD, then that is the highest resolution we can get, 30 megapixel. You can lower it, but you cannot go beyond that. So in our case, in this terahertz reconstructive mechanism, we have eliminated that focusing lens as well as the CCD. And we have incorporated a 3D scanner or nano scanner because it has a nanometer resolution. So we can digitize an object in 3 dimension and then take the digitized data and generate the image. So pretty much the same kind of thing, except that now we can control the resolution. User can select the resolution they want. And the other thing is, because it is terahertz, so we can generate 3D image, not only of one surface, but we can go deeper and scan surface by surface by surface to get a volume image. So the pixel size, as well as in 3 dimension, as we call it a voxel size can be adjusted by user. And it lets us see very small objects. So here's the basic principle of the intensity-based contrast generation is the modified Beer-Lambert's law as we write it in terms of the intensity. And the reproducibility of the intensity comes from the fact that every material has a characteristic reflectance or transmittance. So either way, we can get the contrast. The simple procedure is outlined here that you don't have to worry about now. So as a test of our technique, we just tried to take a cosine function and made a 3-dimensional plot to see if it looks like a cosine function, etc. And the one other thing to say is that the 3-dimensional scanned data, as you can see, the raw data looks like a pile of dots. It doesn't give us a pattern. So we have the special algorithm to convert, much like a camera, convert the intensity matrix into an image. So with this, let me go through how we do this early detection of skin cancer. I mean, it can be applied to other form of cancers down the road, but at the beginning, we are doing this. Skin is the biggest organ of one's body, and therefore the importance is here. So when we image the skin, on the left, as you can see, regular cellular patterns are seen. Those dots are, each of them are the cells separated by all different kind of thing in the tissue. But cells are seen intact. I have better picture later. But when the skin has some kind of a cancer lesion, then the regular cellular pattern deteriorates. So this gives us the biggest opportunity that we can see when cell is healthy or skin skin cells are healthy and when they're not. So here is an installation of the system at a medical college facility at Rutgers University, and this group of medical students and doctors helped us for generating some of the cancer-related data, particularly skin cancer-related data. So before I go to that, as you know, the skin is a layered structure. On the left, see the anatomy of the skin. You have the very topmost layer, which is a stratum corneum, and then you have epidermis, dermis, etc., different layers. So what it means that when you do a scan of the skin from top towards the interior, then we see that it's a layer by layer. This layering property can be quantified. So each of this layer shows in this thickness profile. This is the thickness. So we have 1.2 micron, and we can see the biggest layering between 200 micron and 200 micron and a millimeter. However, this is the case of healthy skin tissue. But when you compare that healthy skin tissue with some sort of a cancerous tissue, for example, here is the basal cell carcinoma, the blue curve on the rightmost, the layering definition has diminished a lot. So this is another way of looking at a skin tissue, either in vivo or ex vivo. These experiments are all done ex vivo. One should be able to see this layering definition. So when and this layering definition measurement doesn't take very long time, so we can quickly see if there is anything going on, or is this skin supposedly healthy or non-healthy. And also the other piece of information we get is from the spectrum analysis. Remember, this machine is a spectrometer as well as an imaging machine all in one. Therefore, here is an ex vivo sample of skin, and that was spectrum was taken. Particularly, this is an eigenfrequency absorbance spectrum. And we see that the healthy skin has a different spectrum or absorbance peaks compared to the basal cell carcinoma skin. So this is another way of quantifying. If we take BCC or squamous or melanoma, different kind of skin cancer, this spectrum will be different, and we can quantify that way as well. So with that, now let me show you some of the images. This is also a basal cell carcinoma on the right side. This is 3D image on the left of a healthy skin biopsy, and on the right, a basal cell carcinoma biopsy. As you can see on the left, again, a regular cellular pattern is intact in healthy cell, but on the right side, the basal cell carcinoma sample has cell on the onset of cell agglomeration, which means that it could form a tumor or a cancer. So, and then by this technique, then we can see a real 3D, meaning starting from the top surface, we can keep on going down as a few millimeters as up until to the bone, and then we can dissect the images, not the patient, but now that we have the three-dimensional image, we can see them one layer at a time. For example, here's the, one of the sample animation. On the left, this animation, as you can see, is not very close, but still in about in 1 millimeter. We only have captured only a few slices, but we can capture as many slices as necessary, and a physician, a doctor, an expert in this area would be able to sit on the computer and see as closely as possible. And now here is another effort where we actually have taken cancerous sample from various patients with the help of those wonderful doctors and medical students you saw. And then we tried to understand difference between different kind of cancers. Out of curiosity, on the leftmost image, in this one is just an image of a piece of gauze, for bandage. We just wanted to see if our technique can see the thing for what it is. And then, there is this sample is a 3D image of a normal skin or healthy skin, where you still see the, cellular pattern. The middle one, number C, is the 3D image of basal cell carcinoma biopsy. And on the right is the squamous cell carcinoma biopsy. So 3D, but different thickness because of the way the samples were taken. And what they did is, this group, as you can see, published here, they have taken about 20 samples of different patients and identified their age, their sex, and from where the biopsy was taken, as you can see, from different places of the body. And then pathologically, they diagnosed it for, or clinically diagnosed for whether it is a BCC or healthy or squamous cell or melanoma. And then they imaged those diagnosed biopsy samples with the instrument. And so here's the instrumental setup. As you can see, a nanoscanning spectrometer. And here are the example of images. So on the top left, this one is a 500 micron by 500 micron healthy skin sample. And this one is 500 micron by 500 micron by the basal cell carcinoma. And when you zoom in, like here, which is a 20 micron by 20 micron healthy skin, so we can see the collagen and that kind of structure of the skin. But as you can see, in case of basal cell carcinoma, the structure has totally deteriorated. So here is a summary of different six phases of 3D reconstructions of a healthy skin. Row 1 is, these are all healthy skin. And row 2 is a basal cell carcinoma. So just looking from different sides, you still can see the healthy cells. Healthy skin has regular cellular pattern that one can see. And the basal cell carcinoma has totally changed the cellular pattern. Here is another comparison with the healthy on the left row, left column. the basal cell carcinoma on the middle column, and the rightmost column is the squamous cell carcinoma. So in summary, now novel terahertz 3D constructive imaging system utilizing terahertz radiation or T-ray that demonstrates non-invasive diagnosis of skin cancer. The reconstructed images display patterns that can differentiate between healthy and diseased skin. It is found that the healthy skin images exhibit a consistent identifiable pattern. The images of basal cell carcinoma and squamous cell carcinoma skin specimens that incorporate clinical photos and histology alongside terahertz images may be able to correlate differences to known pathological features. For terahertz imaging is a useful tool. Therefore, terahertz imaging is a useful tool, a diagnostic tool, and it can differentiate between malignant and benign tissue. Accurately distinguish a scanned image in vivo in a reasonable amount of time. So in vivo image, I haven't shown here, but they did an experiment there too. So now let me move on to the stratum corneum imaging. This experiment was done. So on the left, you see the setup for the college reference cell setup fitted into our spectrometer. And so there is a reservoir at the bottom shown by this yellow, this one, the reservoir. And the top portion where you apply the sample. And then because it is inside the spectrometer, the measurements is being done in transmission. So what happens is when I have this baseline came from just the stratum corneum mounted in this French cell, nothing is happening. So at this point, we have added dilute chromium solution in water, in DI water, onto the top of this, cell of this French cell setup. Then the idea is that if chromium penetrates through this stratum corneum, then the intensity, transmitted intensity, will record that. So as you can see, as soon as we applied that chromium solution, intensity, this is transmitted intensity, although it shows here reflected. The transmitted intensity went down, and eventually it's saturated after a while. So that means all the chromium that can go through the stratum corneum has gone into the reservoir, and some of them are now stuck on the stratum corneum. So we took these two sets of stratum corneum. One is no chromium was applied, and the other one, after this saturation of the chromium chromium solution experiment, and imaged them to see if we can see the differences. So it's a big picture piece of human stratum corneum that didn't have any, no chromium experiment was done. And on the right, that has the chromium experiment was done, both in 3D as well. So on 2D is 1 millimeter by 1 millimeter. On 3D is only 20 by 20 by 20 cubic micron. So we are trying to see what kind of contrast between these two can be imaged. So what we find is that, I don't have a scale here. This is a scale. On the left, the stratum corneum that was not treated with chromium. On the right, chromium treated. stratum corneum, which reflects a lot more light. All the imaging is done in reflection. So it is reflecting more light because a lot of chromium has got stuck on the surface. And that we have seen all along. Even we can come to very close up, such as 10 micron by 10 micron in this case, in this image. And then that gives us a way to quantify the effect of chromium on stratum corneum. So this is just an academic exercise. This is has no practical implication, but it shows that this technique is able to discern between a stratum corneum that was treated with chromium salt and that was not treated with chromium solution. The next topic here, this is another one, is that very much the same way, except that we used special kind of protein to interact, to find out how they interact with the stratum corneum. So on the left side, on the top left side, the stratum corneum that was not treated, and on the right side, the stratum corneum, untreated stratum corneum that was zoomed from 1 square millimeter to 100 micron by 100 micron. So the collagen-like structure comes visible. On the bottom panel, on the left is the stratum corneum treated with DI water. Just deionized water was applied on top of the stratum corneum. And as you can see, the collagen structure on the right, on the top untreated and the bottom is saturated with DI water, that changed the texture of the stratum corneum. So now, the upper left is the protein which they call factor 12. And this protein helps coagulation of blood. So that's where the interest is. So on the upper left corner, this picture here, this image here, where the stratum corneum was saturated with FX2 or factor 12. How we know it was saturated, because we took kinetics, and that data are not shown here. But the time-dependent change in the stratum corneum was recorded. And when data saturated, then we knew that all the factor 12 has saturated the whole stratum corneum. So, you know, the texture of the image on the left is the factor 12. And the image on the upper left here, this one is the stratum corneum saturated with another thing called UM8190. So they show that different protein is having different kind of effect, even over a bigger area, such as 1 square millimeter. On the bottom panel, then we took the close-up of both of those structures, and we see on the 100 micron by 100 micron area, the stratum corneum clearly shows that it's a kind of a structure that you expect from the stratum corneum. But on the right side, when the UM8193 has gone into the stratum corneum, that also showing that there are other material on top of it. It's not just the pattern. So the conclusion here was that that the Cameraless Terahertz imaging measurements of the conformational changes that occurred upon binding of special proteins, namely factor 12 and UM8190 on human stratum corneum. Previously, terahertz scanning reflectometry and terahertz spectrometry was used to demonstrate that different spectral density deviations of the signal are due to the structural alternations and activation of factor 12 induced by the hydrated stratum corneum and UM8190. So that data was published in a different paper, and that's why the spectral analysis is not presented here. But that was the finding. The present terahertz imaging demonstrated that a subset of zygomatin factor 12 bound to the stratum corneum. Factor 12 was not significantly accumulated on the hydrated stratum corneum. However, formed activated factor 12 was observed on the stratum corneum. This finding suggests that negatively charged surface is not necessary condition for factor 12 to bind to the stratum cornea, because there was a theory that the surface need to be negatively charged for this coagulant protein, which is factor 12, or activated, meaning when it is attached, is necessary, but stratum corneum in our case was not charged, as just a neutral surface, and so negatively charged condition is not necessary. Further, the present work also established that the Cameraless Terahertz imaging technique is effectively useful for soft tissue imaging, both on the surface and under the surface. So final conclusions are, it is a no-brainer that early detection of any disease gives a better treatment option for the physicians. The skin is the most affected organ by cancer because a great majority of the cancer is skin cancer. Basal cell carcinoma is the most common skin neoplasm. Squamous cell cancers may be preceded by actinic keratosis. Keratosis, forgive my pronunciation, keratosis, premalignant lesion may invade deeper structures and metastasize, and so we can image deeper and find that out. Aggressive local growth and metastasis are common features of malignant melanoma, which accounts for 75% of all deaths associated with skin cancer. Therefore, early detection greatly improves the prognosis of patients with malignant melanoma. It is non-invasive, non-contact terahertz imaging, spectroscopy, and profiling, all of them together have diagnosis of skin cancer at very, very early stage. And this combination of these three techniques improves the chance of diagnosis with, false positive and false negative. Because when it comes to diagnosis, we don't want either false positive or false negative. If it is the false negative means that if the disease is there and we couldn't diagnose it. And false positive means the disease is not there, but yet the machine says it is there. So we don't want any of those cases. Therefore, the combination of these three techniques in one machine, which is imaging, spectroscopy, and thickness profiling, improves the chance of eliminating a false negative and false positive. And maybe extended to the genetic level diagnosis, because that's a different story I didn't go through over here. And what does it take for us to use it as a commercial diagnosis tool? It is, we need to study more patients and then apply for FDA approval. So with that, I thank you for your attention and happy to answer if you have any questions.
Thank you so much for that lovely talk. Are there any questions? If not, I have a quick one. I'm wondering about the cost effectiveness of having this kind of testing into the healthcare system, or is it quite expensive?
Well, you know, instrument is a bit expensive, but access shouldn't be. So if you install this instrument, what do we want? We want the patients should have access, easy access, and quick diagnosis. So there is no, you can get immediate result. This instrument can be installed, let's say, on a dermatologist's desk or a doctor's desk, or even a centralized position in a hospital or clinic. There is no consumables. So,
yep.
You know, it's not like I have to have every day some replacement or something like that. There's no consumable here. Therefore, it's a radiation. We have built this machine 7-8 years ago, and still we are working with it. And you know, our company offers 2 years warranty, but machines go, you know, 6, 7, 8 years without any difficulty whatsoever. So what I'm trying to say is that the access to the patient is the most important thing. So let's say machine costs $100,000, but that cost is not going to be transferred to the patient because of this longevity of the machine, no consumables needed, and seamless operation of the machine. In fact, our second generation of this machine will be to do a whole body scan with terahertz radiation. So you can incorporate that, include that in an annual physical, for example, to see if my skin is healthy or not.
Okay, thank you very much.